Welcome once more to this YouTube channel. Kindly subscribe to this channel. The topic is biochemical genetics, a vital topic in medical genetics for biomedical postgraduate students, that's the PhD students. What are the objectives? Candidates should explain how a collection of mutants with the same phenotype can be used to study a biochemical pathway. They should know how to identify mutants with mutations in the same pathway and how to characterize the mutants into complementation groups. Candidates should know how to use epistasis tests to infer details about a biochemical pathway. They should know test of dominance. They should understand test of complementation. They should cognize test of epistasis. They should know the molecular basis of common biochemical disorders. They should have an understanding of the genetic causes of inborn errors of metabolism, inheritance patterns, and heterogeneity. They should know the biochemical techniques and techniques used for the diagnosis and management of inborn errors of metabolism. Candidates should have an awareness of treatment options and dietary management of metabolic disorders. Right here is an illustration of epistasis, which is the interaction here between hair color and baldness. Baldness, the genes that is responsible for baldness tends to mask the manifestation of hair color such that the individual will end up with red or blonde hair color. So there is a form of interaction between the genes that causes baldness and hair color, that is epistasis. What are the topics? First topic is the mechanism of inheritance of metabolic disorders. The second topic has to do with gene action. The candidates need to be taught the analysis of changes which occur in phenotype when a gene undergoes mutation. And they also need to be taught the determination of the direct effect of genes over metabolism. Number 3 topic here is heterogeneity. That's genetic heterogeneity, where you have mutations at 2 or more genetic loci that produce the same or similar phenotypes, either biochemical or clinical. You have the same phenotype from different mutations occurring as well. Then you have clinical subphenotypic heterogeneity, where you have different mutations in the same genes that can sometimes give rise to strikingly different phenotypes. You have different phenotypes from the same mutation in the same gene. Example, you have dwarfism and gigantism. The fourth topic here is mechanism of pathology. You could have acclimation of point mutations. So the genetic mutation could be as a result of acclimation of point mutations. It could be caused by insertion or deletions, causing frame shifts. You could have changes to essential regions being modeled. All these are forms of genetic mutations that can result in pathologies that affect the biochemical pathways. The fifth topic here is metabolic disorders. You look at dysfunctional protein causing a block in the metabolic pathway, and this leads to acclimation of enzyme substrates. Also, you have formation of unusual substrates and then deficiencies in product. Now, categories of genetic errors is another topic. You consider amino acid metabolism, where you look at phenylketonuria, for example, carbohydrate metabolism and the glycogen storage disorders. You could also have disorders of fructose metabolism, disorders of galactose metabolism, where you have galactosemia. Then diabetes and hypoglycemia is some other category of genetic errors that could result. You could also have disorders of the urea cycle. You could have disorders of purine metabolism, disorders of pyrimidine metabolism, that's the porphyrias. disorders of beta oxidation and electron transport chain, these lipidemias, disorders of cholesterol and bile acid synthesis and storage, organelle functions, glycolipids and complex carbohydrates, lysosomal storage disorders and other organelle function disorders. Organic acid metabolism could also be affected, where you have medium chain acyl coenzyme A dehydrogenase deficiency, and also you could have neurometabolic disorders. Peroxisomal disorders are also there, disorders that affect heme biosynthesis, like you have acute intermittent porphyrias as well. Then you could have GM2 gangliosidosis, where you have the Tay-Sachs disease and the Sandhoff's disease. Right here is an illustration of the GM2 gangliosidosis. Here there is mutation of the hexa gene, that's the hexosaminidase A enzyme is affected. There is mutation of the hexa gene, and this results in deficiency of hexosaminidase A. You have accumulation of GM2 in the ganglioside in neurons, and this results in membranous bodies inside the neurons. There's neuronal degradation, demyelination as well. You have cherry red spot in the macula of the eye, and narrowing of the blood vessels, and the patient manifests with macrocephaly, hypotony, mental and motor delay, and then convulsions. So this is a metabolic disease. Check out other videos on this channel for details of all these topics. Don't forget to subscribe to this channel. Thank you for listening. Goodbye for now.